All right. All right, so let's talk about red blood cells. Your RBCs. And we're just gonna, let's just draw a little one from the side. So they kind of look like this. From the side. They're a little biconcave disc. So rather than just being a disc like this, they kind of go in the middle. If we draw one kind of face on, they look something like this. You got like a little divot in the center. Let me pull up a little picture of one because my drawings are not doing justice, of course. So this is a little electron micrograph of a red blood cell. You can see they got this little divot. It's called a biconcave shape. And this allows them to be pliable and bendable so they can fit through your capillaries, which are about one red blood cell in size. All right, let me just, I don't know, I just want to spit some facts. So we're going to, I mean, this stuff is crazy. You're made out of, you're made out of about 37 trillion cells. I've told you that before. You are made out of about 37 trillion cells. That's kind of crazy. 37 trillion. That's a lot of zeros. I mean, let's just do it. 37, 0, 0, 0, 0, 0, 0. That's 37 million. That's 37 billion. That's 37 trillion. Thousand, million, billion, trillion. Okay. About 70% of your cells are red blood cells. That's crazy. Okay. Y-O-U-R. All right. And they live for about 120 days. So 70% of you is completely different every, what is that, 4 months? So 4 times a year, 70% of you is completely different. And other parts of you are also completely different. But if you think about, if you think about that, it's really kind of crazy. You're mostly blood, and you're mostly red blood cells. Are mostly red blood cells. As far as cells go. They're the most abundant cell type. That's really kind of bonkers to me. We'll talk about this in the next lecture, but 2.4 million red blood cells are made per second. What? 2.4 million red blood cells are made per second in your bone marrow. That's crazy. Okay. Really, that is bonkers to me. And why is this? What do red blood cells do? What do red blood cells do? Well, we talked about it already in the last one. They carry our gases. Oxygen and carbon dioxide. Okay, they carry our gases, oxygen and carbon dioxide. How do they do that? Well, here we've got a red blood cell. Let me redraw a red blood cell here. I'm going to draw it in purple on the outside. And let's see how well I do this. This is a red blood cell. It's a red blood cell. Let's see how well I can do this. I'm going to bring over a David Goodsell drawing. Boom, like this. All right. So I've lined up this membrane with our red blood cell. Red blood cells do not contain a nucleus. Very interesting. They don't contain a nucleus, and that's because they want to have this biconcave shape. If they had a big fat nucleus in there, it'd get in the way. These boys got to pack in tiny. They got to be flexible. So that biconcave shape, no nucleus to allow for biconcave shape, to allow for flexibility to fit into small spaces and to be able to maintain flow. Smally spaces, flexibility. Very important. Okay. Let me pull up a little example of this, why this is important. There is a little disease called sickle cell anemia. This is caused by a single mutation in a protein in hemoglobin, which we'll talk about in a second. And what this gene does is it causes your cells to go from this nice flexible disc-like shape to like this little sickle shape. Looks like a sickle that you like cut wheat with, or like a little banana. And this is a very painful disease. What it results in is your blood cells basically getting stuck together in your capillaries. So these little bananas get all stuck together and then block the flow of blood. They're not as well as good at carrying oxygen, and they also sort of gunking up the works here, gunking up the pipes to kind of slow down your cardiovascular system, prevent the delivery of oxygen and all that. Okay, so the main function of a red blood cell is to deliver oxygen. The main function of a red blood cell is to deliver oxygen to cells, to feed that mitochondria, to cells, to feed the mitochondria. To feed mitochondria. That's the main function of a red blood cell. How does it do that? Red blood cells contain hemoglobin. Hemoglobin. Edit. To carry oxygen molecules, to carry oxygen gas. All right, and that's what we're looking at here. So here we have the plasma membrane of a red blood cell. Here we have the plasma. Look at these little antibodies. You can see these little antibodies here. Out in the plasma, we got the membrane. And then inside all of these little looking things, they look like just little globs. All the red things, these are all hemoglobin. So let's zoom one of these out and just kind of draw very generally what it looks like. Well, I'm just going to draw this from memory. Doesn't have to be perfect. So this is a protein. It's made out of 4 subunits. I'll draw it like that, and then one like this, and then another one like this, and another one like this. All right, so that's one hemoglobin molecule. And these guys they have 4 heme groups, boom, 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 boom. And those are made of iron. So this is one hemoglobin. And this, let's go in here. This is a heme group, and it contains heme group. We'll leave it like that. Heme group contains an iron molecule. A metal, Fe. That's the atomic symbol for iron, which binds to oxygen. Well, think about rust. Just like oxygen will rust iron, or steel. Steel is just made out of iron, special heat and all that stuff. Makes steel versus iron, but iron is steel. Or I'm sorry, steel is iron. Just like oxygen will rust iron, oxygen loves to bind to iron. Okay? So these will carry our little oxygen molecules, these little groups. So each hemoglobin, and hemoglobin is a protein, it's polypeptide. Hemoglobin is a protein. It's made out of 4 proteins, 4 subunits that come together in a tertiary structure. Each hemoglobin can carry 4 O2s, 4 oxygens. Oxygen gas can carry 4, F-O-U-R, 4 of them. Okay? You have about 250 million of these in every red blood cell. It's pretty crazy. You have about 250 million hemoglobin in each red blood cell. Each can carry 4, meaning 4 oxygens, meaning your each hemoglobin can carry 4 oxygen, 4 O2, oxygen gas, meaning each red blood cell can carry about 1 billion oxygen molecules. Okay? That's crazy. You take that 1 billion times it by this, 70% of 37 trillion, like, I don't know, 20 something trillion. The numbers get crazy here. But it's a lot. It's a lot of freaking oxygen. This thing's like an oxygen bank. With all the money in the world, it wants to just hold that oxygen. It's crazy. Okay? Some like little fun facts here. I got this over here on it. I think I already put it in here. Yeah, I did. Okay, so I got this diagram here. This is pretty cool. We can see what a better artist rendition of hemoglobin looks like. So this is hemoglobin. These are these little heme groups with these iron atoms that will hold our oxygen. Okay? And your red blood cell has 250 million of these in them. Each one has 250 million hemoglobins. It's crazy. It's absolutely crazy. There's also something called myoglobin, which is in your muscles. So this is another oxygen-carrying molecule. It doesn't have 4, it rather just has 1. You can kind of see it's pretty much just one of these on its own. And this allows your muscles, this is stored in your muscles, to hold on to some oxygen, because your muscles are using oxygen all the time if we're really working out. And this gives red meat its kind of darker color. Dark meat, it's dark color. It's because they'll have more myoglobin. So something like fish has very little myoglobin, chicken kind of in between depending on the meat, and then something like beef has a ton of myoglobin. And this is why meat, like beef, has iron in it. It's because of the myoglobin, bruh. Get with them times, son. Learn a little bit about the food that you're putting in your belly. I think that's pretty cool. So as your blood is whizzing by your muscles, if you're exercising, not only can these hemoglobins deliver oxygen to your muscles, but they can also deliver some to this myoglobin in the muscle so that you can have this extra store, this extra buffer of oxygen storage in your muscles through myoglobin. Pretty nifty indeed. Okay, one last little thing that I want to talk about with just the general kind of shape of, and this should actually go up here. I think this is really cool, like a cool physiology thing. Why can camels go such a long time without water? Well, this is one of the reasons. There's multiple. But human red blood cells have this biconcave disc shape. You can see they look kind of circular. You can't really see the biconcaveness here too well. A little bit. You can see it's like lighter in the center, but they're circular. Whereas a camel cell is more like a football or like a pill. It's longer like this. And when it's longer and stretched out like that, when you drink a bunch of water, it can change its shape more, right? Because it's already kind of stretched out like this. Whereas something like this, it can only change its shape that much. It can only absorb that much water. Whereas a camel cell, like our blood cells, they're like this shape from this, the side. It can only absorb that much water before it like bursts. Whereas a camel's blood cell is like this, so it can absorb a lot more water and hold on to it. They're elongated. What's up? What's up? You get it? That's pretty nifty. So that's one of the reasons why camels, they can gorge themselves on way more water, and they store it in their red blood cells. It's nifty. I think that's really nifty. Okay, let's keep on cruising. Where are we at with this? All right, we're going to keep on going. We're going to talk about what is blood type, because that has to do with your red blood cells and your plasma. What is blood type? What is blood type? All right, let me pull this up here. I just need a little reference to draw from. All right, so we got a red blood cell. So what are our 4 blood types? We can have A, we can have B, we can have A, B, and we can have O. Those are our 4 blood types. Come on, baby. What are we doing here? What's going on? My life is falling apart. Okay, boom. A, B, O. A, B, O. Okay, I am O blood type. It's the most common blood type out there. What does this mean, though? All right, well, in all of these, we got red blood cells. We got red blood cells, red blood cell, red blood cell, and a red blood cell. Okay, what does this mean? What does this mean? What does this mean? Well, the type of blood that you are is talking about the surface proteins. So, for example, an A blood type, let's say it's got these like rounded surface proteins, something like this. So, we got some surface proteins up here on this picture. So look at all this stuff. We got all these surface proteins. These are glycoproteins, different shapes, right? We got the membrane, and we got these proteins on the surface, all right? That's what we're talking about. Different shaped surface proteins, all right? And A blood type, let's say it's got these rounded surface proteins, something like that. Okay, B, let's say this bad boy got a real spiky looking surface protein, something like that. Something like that, all right? AB, it's gonna have both of those. It's gonna have these spiky boys. That one's a little tall, it's okay though. And then it's gonna have these roundy boys. Like so. Oh, oops, we forgot them. Doesn't have it. No surface proteins. No surface proteins. Pretty special in that case. No surface proteins. We got round surface proteins, spiky surface proteins, spiky surface proteins, and then a combination of both of these. We got round surface proteins and spiky surface proteins. That's what blood type means. Okay. What happens though? So in our plasma, what do we have over here? What were all these things? These are called antibodies. And these recognize things. They're like little specific shapes. And they bind to specific things. So in our plasma, so these are our red blood cells. Red blood cells. And this is in our plasma. Plasma. We will make antibodies. So your blood type A, if you're blood type A, you make antibodies that will recognize this bad boy. So your antibodies will look like something like this. They'll have that reverse spiky, and then they'll have that kind of Y shape. All right. It's not the not the best thing I've ever, not the best antibody I've ever drawn. Maybe I'll draw this one better. Okay, if you're B blood type, I'm going to draw it in gray, just so it's also a different color. It's going to recognize this. So it's going to have little rounded. I need to probably zoom in just for. It's going to have little rounded, and then Y. Okay, that one looks better. Still a little goofy, but you get it. Okay? And if we're AB, no antibodies. No antibodies. And if we're O, you actually make both. You make both of these bad boys. I'll put them over there and then over here. Make both of these. Beautiful. Okay, so let's say we, all right, so our A type, it has to do with our surface proteins. And we basically make, so A makes something that binds to B. It makes an antibody that binds B. We'll just put it like this. Binds to B. A binds to B, which means B, right? This binds to A, binds to A, binds to nothing. There's nothing to bind, right? And then the plasma here binds both A and B. Pretty nifty. So whatever you have on your surface, you have the opposite in your plasma. That makes sense. So here we have both, so we have nothing. Here we have nothing, so we have both. Here we have A, so we have B. Here we have B, so we have A. All right, now let's just take an A red blood cell and mix it with B plasma and see what happens. So we're going to do a blood transfusion between somebody with A blood and B blood. Okay? All right. So what we're going to do is we're going to add, so we got somebody with B blood here, and we're going to add A red blood cells to it. I'm going to copy these, bring them down. So we're going to do a blood transfusion by adding A red blood cells to somebody with B blood. What's going to happen? So we're going to add in a bunch of these red blood cells. They're going to bind like so. This is not good. We don't want this. This is very, very bad. Did my best here. Trying to get this nailed down. I'm going to end up moving that surface protein, but that's okay. I'll just get rid of it in a second. Okay, and let's just copy this over here so that we can kind of complete this little chain. What we're doing here, if you can see, is we're essentially coagulating all of our blood. We're glutenating it, it's called. And we're aggregating all the red blood cells together. This is very bad. This would not flow through our bloodstream. In fact, this would kill us. We would then die. It's essentially like we're giving ourselves an extreme version of sickle cell anemia or something like that. So this is called agglutination. So we took this and we did a transfusion of A blood cells into a type B person. That's what we did. We did blood transfusion. This causes agglutination. Agglutination or aggregation. We made an aggregate. We've bound aggregation or bound red blood cells together with antibodies. That's what this thing is called. And I don't know if I said this. This is called a plasma antibody. Antibodies. If you want to know a pro tip here, I don't care that you necessarily know this, these things are called antigens. Things that an antibody recognizes are an antigen. All right? So that's what the fuss about blood type all is. All right? About the letters A, B, and O. All right, one more thing that I want to talk about, which is the RH factor. And this is called where the minus comes in. So RH factor. So this is, if we have O, let's say we have A positive versus A negative. What does that mean? So A positive, there's just another surface protein. Let me grab A and bring it down here again. So this would be A positive, or actually this would be A negative. It doesn't contain the RH factor. I'm not sure what just happened here. Let me pause this. Okay, sorry about that. Sidebar pop up, didn't know how to shut it off. Agglutination, RH factor. Okay, so our RH factor has to do with this. It has to do with that. What that means. For example, I am O positive. That's what RH factor is. It has to do with this, plus or minus. Okay, so a minus doesn't have this other additional RH factor, surface protein. Whereas positive would. So let's just draw it as like a little, let's draw it as these little arrows. Looks pretty nifty. But it's the same kind of thing happens here. All right, so has RH factor surface proteins. Okay. Has RH factor surface proteins. All right, so if we are RH positive, we don't make antibodies against this. So this would be, well, I'll also draw it in green. It might be hard to draw this little thing that recognizes an arrow, but it's okay. So if you're A negative, you do make, you guys will get it, this will recognize those little arrows. You do make antibodies that will recognize RH factor in your plasma. So this is being the plasma. This would be red blood cells. Red blood cells. Okay? So this will bind to this and aggregate the blood. So if you're negative, you make Rh positive antibodies. Okay, and this is pretty interesting. So if we take, because this has to do with pregnancy. So if you have a mother who is Rh negative. So we got a woman who's Rh negative, and we've got a father. I'm trying to not be gender specific. I made that in pink, so I might as well just do it. I don't like gendering with blue and pink, but I did it, okay? Son of a bitch. Well, I'm so heteronormative. All right. We got an A positive, let's say. A positive daddy. And an A negative mommy. And then let's say, boop boop, she gets pregnant with an A. She's pregnant now with an A positive baby inside of her. This is bad. This is really bad. Because she's going to make a bunch of these antibodies. She's going to make an immune response to these antigens. So these aren't normally actually present. So plasma will make anti-Rh factor antibodies. Your immune system, if they are present. Okay? So if they are present, your immune system will make these. And so this isn't necessarily a big deal for this baby. It's not necessarily a big deal for that first kid. That first kid will be fine. But what happens is some red blood cells during pregnancy or like right after birth will leak into the mother. So red blood cells will leak into the mother. During pregnancy or after pregnancy. Red blood cells from the baby will leak into the mother's bloodstream from the placenta. A couple of them will slip through. Placenta. Then the mother will make a bunch of antibodies against it. Make a bunch of antibodies to detect it. The mother will then make a bunch of antibodies against our H factor. Surface proteins. All right, and this is a real then problem if she has another A plus baby. Let's say another Rh factor positive baby. If she has a second, if pregnant with a second A plus Rh factor positive baby, or B plus or O plus, whatever, she has it with a different dad, doesn't matter. Her immune system will attack the fetus and kill it. Because this process is going to be happening, this whole thing. So how do we prevent this? So to prevent this, we inject the mother with Rh antibodies. It's pretty nifty. So we inject her with these. So if any red blood cells from the little baby leak in, they will cover. So let's say this is a little red blood cell for the baby. And then we have a bunch of these antibodies. We inject it into the mother a couple times during the pregnancy. Really kind of, I think it's just twice. Like around 28 weeks and then like right after birth. And the reason why we do this is this will cover up these antigens. It'll mask them and hide them. So her own body will not make antibodies. Because these are all now hidden. It's a pretty nifty little trick that we do. I think it's really cool. So those are all now hidden. Injected Rh antibodies hide the Rh surface protein, preventing the mother's immune system from making buttloads, many loads, making many antibodies that would then attack and reject a second Rh positive child, fetus. What? It's pretty cool. I think that's really cool. Hopefully that made sense. But that's what that plus and minus is. It's just another surface protein. But you're not normally making a bunch of those antibodies. Okay, I went on a little long talking about Rh factor with this one. Pretty cool stuff though. Red blood cells are pretty fascinating. Okay, let's talk about white blood cells.